Hello everyone. I am delighted to share with you our recent publication in iMeta titled Soil-borne Legacy Facilitates the Dissemination of Antibiotic Resistance Genes in Soil Plant Continua. First, why is this issue so important? And how is it connected to each of our health? The transmission of ARGs from soil to crops represents a major health concern. Disease-suppressive soils promote plant health, yet paradoxically select for ARG enrichment through antimicrobial compounds produced by beneficial microbes. Highly resistant pathogens, such as Ralstonia solanacearum, further modulate soil resistance. However, the mechanisms by which such pathogens drive ARG transfer across the soil-plant continuum remain poorly understood, limiting our ability to predict resistance flow into crops and human populations. To address this knowledge gap, we investigated bacterial wilt caused by R. solanacearum in a tomato greenhouse with a long history of disease, where the infection subsequently spread to adjacent strawberry crops, revealing a persistent soil-borne legacy. We hypothesized that R. solanacearum infection enriches specific ARGs, which persist and carry over to subsequent crops even in the absence of visible symptoms. Using high-throughput qPCR and shotgun metagenomics, we quantified shifts in resistance and microbial communities and identified ARG and MG-bearing pathogens, thereby evaluating AMR dissemination risks in agricultural production systems. Our analysis demonstrates that soil-borne diseases, including bacterial wilt, actively drive the dissemination of ARGs within agricultural systems. This process establishes a persistent ecological legacy, facilitating the transfer of ARGs to subsequent crop cycles. Importantly, these ARGs frequently co-localize with MGEs, highlighting a direct mobilization risk to human-associated pathogens. Invasion by R. solanacearum reduced microbial diversity and reorganized plant-associated microbial communities, thereby weakening ecological stability. The resulting pathogen pressure intensified antagonistic interactions, fostering the recruitment of beneficial and antibiotic-producing taxa and inadvertently enriching ARGs. These infection-driven shifts indicate that microbiome restructuring constitutes a central ecological mechanism that amplifies resistance across soil-plant continua. Metagenomic analyses revealed that healthy plants harbored the highest diversity of ARGs, predominantly multidrug, bacitracin, polymyxin, and beta-lactam resistance genes. The rhizosphere emerged as a hotspot for ARGs, MGEs, and VFGs. Both beneficial microbes and pathogen infection contribute to enhanced HGT and ARG enrichment. ARGs disseminate through atmospheric deposition, HGT in the rhizosphere, and soil fauna, with R. solanacearum serving as a major ARG reservoir, demonstrating that both healthy and diseased agricultural systems substantially expand the environmental resistance. Most ARGs detected in the fruits originated from the rhizosphere of healthy tomato plants, revealing a silent, cross-crop transmission pathway. Microbial interactions and selective pressures enrich ARGs and facilitate their MG-mediated dissemination, thereby expanding the soil resistance and enabling resistance transfer across the soil-plant continuum into the food chain, thus posing potential risks to public health. Metagenomic analyses revealed frequent colocalization of ARGs, MGEs, and VFGs, enabling direct HGT between soil microbes and R. solanacearum. Beneficial biocontrol microbes can act as ARG reservoirs, while pathogens can currently acquire clinically relevant resistance traits. Notably, vancomycin resistance genes were transferred from soil into strawberry fruits, underscoring agricultural soils as critical gateways for ARG entry into the human food chain. Our findings demonstrate that R. solanacearum promotes ARG persistence and dissemination across the soil-tomato continuum, even in the absence of visible disease symptoms. Soil-borne legacies further facilitate ARG transfer into subsequent crops and edible plant tissues. Key limitations include the constraints of short-read sequencing, uncontrolled environmental variables, cross-sectional sampling, and host DNA contamination. Future studies should integrate long-read sequencing, functional assays, and long-term monitoring to resolve ARG-MG host relationships and track resistance dynamics with higher precision.